The topics we will cover in this presentation are basically the rules around out-of-specification investigations, the different phases based on the MHRA and FDA guidelines, and we'll also have an extra section just on outlier tests and how many resamples you might decide to use when you do retesting. There are various useful guidelines for out-of-spec investigations. There's the famous FDA guidelines, and more recently the MHRA have produced some good presentations and methodology for that. And there's the PICS aid memoir to inspection of quality control laboratories. The FDA guidance covers all the chemistry-based testing in the pharmacopoeias and the biologicals as well, but it doesn't cover in vivo biological assays. It includes testing on APIs, excipients, other starting materials, finished drugs, anything you get from contract manufactured organizations, contract laboratories doing work on your behalf, and any of your testing results you get as part of your stability trials and validation studies and monitoring of your critical utilities. You will get out of specifications anywhere in the pharma factory where there are specifications that need to be met. It sounds obvious, but there are obvious exceptions, like during stability trials and environmental monitoring and even production monitoring, where you may have some alert limits and action limits. We'll talk a bit about those next in terms of out of expectation and out of trend, but essentially finished products, starting materials, and so on will have specifications which you can get an OOS for. Before we start, I realize that there's a lot of discussion on the internet, LinkedIn for example, about out of expectation and out of trend results, and some confusion about what they are and what OOSs are. So out of expectation may be an example where there's a batch potency adjustment. So you've got the potency of the starting material and you make an adjustment, but the final result is not what you expected. So that would be an OOE, and you might investigate what happened there. Might similar process to out of spec, but nothing's out of spec yet. Another case may be like a pH adjustment. Again, you've made a pH adjustment to an injectable bulk product, and it's not where you expected it to be. There's no trend to consider, so it's not actually out of trend. And you might get them in out of environmental monitoring as well. You get so some practical examples could be with for example, stability trial, where it's being following a steady trend or not trend at all, just a straight line, and then there's one up and down, a down movement, but it's still within specification. Also in production, might get production processes suddenly behaving different to what they used to be or normally do, but they're still within the alert and action limits. In these cases, production QA, QC may still pull together some investigation plan to find out what is going on. The expectation is that your process is in control and that you don't just ignore things if they're still within specification. The current guidelines split the investigation into 4 stages: 1A, B, stage 2 and stage 3. The first part is the initial investigation in the lab to make sure something obvious hasn't gone wrong, the wrong dilutions or something like that. Stage B is a much more in-depth laboratory investigation. Stage 2 starts to include production and the potential for sampling, and we'll talk about those a bit more in detail following this slide. Here is an overall top-level flowchart of the process expected to be used. So you get an unexplained result. It gets reported to the QC manager. QC manager will do the initial phase 1A assessment, looking for really basic mistakes like you did the wrong sample or knocked it over or something. If it doesn't get past that, it'll go to 1B, which is a more in-depth look at the methodology, the instrumentation, the calibration, looking at validation reports, looking for still laboratory-based issues, which but much more harder to spot. If it goes beyond that, then this is the first time it becomes a combined production and lab investigation, and it's important to understand that this phase is where you do talk about retesting the same sample or potentially new samples, but it's much more complicated, and we'll get into that later. And the phase 3 is a final combined production lab assessment, and that's when you discover whether it's a real OOS and the product is defective and the batch is rejected, or not. And that then goes through the kappa system as usual, and you may have reworks and things like that to do. This is a list of the stages you would go through over the course of the whole investigation. Like I said, during the early phases, it's focused in the laboratory, and there's many steps which then lead you to looking at the batch itself and homogeneity. But the key point in this slide is to show it's a stepwise process, and you don't jump into all different aspects from the word go. As far as any employee is concerned, whether it's in production or the laboratory, the employee should be trained to not, I guess, deliberately create an OOS starting machine when you know the calibration's not right, running a run of samples when the system suitability wasn't right, or you know you made a mistake in a dilution or something. There are quite a few biological assays which are time consuming. For example, the endotoxin test, and there's a kinetic step in there where you have to use a heating clock for a certain number of minutes. It's quite easy for an analyst to make a mistake there and cook it for too long, and you end up with a invalid assay, and causes a lot of waste of time. It's not actually an out of spec, but the main point is don't proceed with assays and testing or production if you know it's not going to work out in the first place. Overall, the investigation should follow a predetermined and structured plan. I mean, that's what the SOP should allow for, and the finished product should be the report, which can lead to product release and any kappa or change controls that are required. The investigation should always look forward and look back, in that if it's an analyst specific or instrument specific, it may be more than one batch that's involved. Look for trends in common products and analysts, other trends in like changes of shifts and things like that, and what is the impact on materials inventory out in the marketplace, work in progress. Once you start digging into these things, the tentacles can go a long way. So now we get into the details. Phase 1, the obvious laboratory error. You know, all your samples and test solutions that you use for testing should be kept irregardless until your calculations have been checked and the results passed by the QC supervisor or manager. You should have enough samples, and the sample size should be enough to cover 3 times testing, and that doesn't include your normal retained samples. So simple laboratory errors, the analysts can get the workbook checked by a senior analyst, and they might find calculation errors. They might have used the wrong standard, or the standard may be expired or close to expiry and not look the same as it has done over 6 months, 5 months ago. There may have been a power failure, there may be like leaks in the piping on the HPLC and so on. So 1A is really looking for basic laboratory errors in microbiology and chemistry, you can basically discount the result on a factual basis. This slide simply shows more examples of the obvious laboratory areas you could look for in the initial investigation. So this flowchart simply is a breakout of the original one in more detail. So the phase one investigation, if you find an obvious error marked on the left in red, you still have to document the quality event in your QC system or the QA system, depending how your company is set up. And if it is a problem with the method, that people are finding workarounds to, something like that, then you might still raise a CAPA to correct that if it's a chronic issue. Examples of that I've had in the past where there's a laboratory method which involved sonicating samples and filtration steps and extraction steps, and all these steps were not really specified precisely in the method, and it was the senior analysts who knew the method from a long time ago, who knew what these, I guess, techniques were, and they had to be put into the method. So it's things like that that you might put a CAPA up for. If there's nothing obvious found, you go to the phase 2B, which is the more in-depth investigation. So if you're unfortunate enough to have to go to phase B and what's called an indeterminate error, that is, you cannot on initial investigation figure out what's the problem is, you have to go to phase B. The OOS should be raised and the QA manager should be immediately informed and the investigation commenced before any retesting or resampling is carried out. That's kind of a difference from what used to happen, is that QC were often left to their own devices to start with and retest and so on, but that's changed, and that's a major change because QA are seen as independent from QC, so QA need to agree to any kind of retesting and resampling. So during the phase 1B, there's the same, I guess, interrogation of the analysts and confirm the knowledge and training records. You've got to also make sure the sample was taken by the right person. Some companies don't have a specified person, but when you have complicated processes, it is important to make sure you get the sample that's representative from the right part of the process, and this is often figured out during the R&D process development stage. Also, you've got to look at all the chromatograms and look at any changes in relative retention times and the peaks. Are they using a new column? Have they just changed columns? Further looks at the instrumentation and calibration. Has the service guy been in recently and changed something and logs not being filled out? So it's a much more detailed investigation. And the idea is for the supervisor to be absolutely sure what kind of category the OOS is for the rest of the investigation. So this flowchart briefly explains what happens when you get to phase B. The QC manager would have to come up with a hypothesis as to what they think's going on with the results, and on the left hand side, if the QC has done all its checks, it checks all its records and everything checks out fine, there's clearly no assignable cause found. The only answer therefore, I guess, is the product. So production investigation is required and phase 2 starts. If the investigation uncovers an assignable cause in the lab, then they can invalidate the test data, repeat the analysis and record a new result and close out the investigation. And they would also have to raise a CAPA to prevent whatever happening again. This slide reiterates what I said on the previous one, but briefly mentions microbiology and not throwing samples out. And at this stage, once the QC results have been reviewed, and you're at 1B and you have got a hypothesis test to carry out, at this stage you can still only do these tests on the original sample or preparations of the original sample. It's ideal if you've got the original volumetric flask, for example, but if you've got the original bottle of powder or tablets or vials, so much the better. It's not time yet to go out in the factory resampling yet. This slide simply has a formal definitions, examples of the types of outcomes you might get in the 1B stage of the investigation.